Great. Welcome, everybody, again to another Inside INmune Bio edition. What I'd like to do today is a little bit different. Kind of given myself a challenge, and what I thought I would do is I'd go through our pipeline chart, and with only using one slide, I would try and describe what our program is and why it makes sense. It's pretty difficult using just one slide, but if you bear with me, let me know in the comments if you think I did a decent job or not, or if there's something else I should have covered. That'd be great. And again, we have another hat here. It's a little bit different. This one's kind of cool. It says INmune Bio on the back. We'll pick one of the better comments and we'll ship you a hat. So share the video, like the video, and we need at least 25 reasonable comments, and we'll pick a random person and send you a hat. So what you see here is our pipeline. I'm going to talk about the 4 active programs that we're developing in the clinic. 2 are around XPro, and 2 around INmune. In XPro, we have our early Alzheimer's program, which is currently in phase 2. It's recruiting patients in Australia, Canada, and we hope to have other venues open in the United States and parts of Europe soon. The second program is around treatment-resistant depression. It's a phase 2 program as well. And I have only one slide to talk to you about why we're excited about treatment-resistant depression. And then switching over to INmune, we have one ongoing program that's currently in the UK and just had a site open in Greece around a form of leukemia called myelodysplastic syndrome and AML, acute myeloid leukemia. And then the second program in INmune is around metastatic castration-resistant prostate cancer, which is a phase 1/2 program. They'll start later this year here in the United States. We're very excited about all 4 of these programs. And let's move on. Let's try and do this with one slide. So let's talk about our Alzheimer's program. You know, we look at Alzheimer's very differently. We know and recognize there's been a lot of failures in Alzheimer's. And part of the reason is really that they've been taking the same approach over and over again. They've been targeting these plaques, taus, and until recently, they really haven't had just great, they've had really terrible results. And until recently, they've had some recent approvals with Leqembi, Aduhelm, and soon donanemab from Eli Lilly. So what is Alzheimer's disease in our view? You know, we view it really as an immunologic disease, immunologic dysfunction of the brain. And when your immune system is dysfunctional in the brain, we really believe you start this degenerative process. And that dysfunction really starts from neuroinflammation. So if you look at the picture on the screen, this is really our simplified view of the driver of Alzheimer's disease. You have 2 things that drive the disease. You have neurons, which store the memory, and then you have synapses, which really allow the neurons to communicate. There's these fiber tracts that allow information to travel. It's what allows me to lift my hand. It causes movement, recall of memory, et cetera, et cetera. And the immune cells of the brain are responsible for taking care of the neurons and the synapses. And these immune cells, such as microglial cells, when they're healthy, they do their job. They remove debris, they take care of the neurons, they take care of the synapses, et cetera, et cetera. And when they're dysfunctional, they don't. So a simple analogy, tried to come up with a good analogy, and we really borrowed this one from CJ, is that the nerve cells and the synapses are like infants. They can't take care of themselves. They need somebody. They need a mother or a parent to take care of them. And those parents are the microglial cells. And the microglial cells do their job. They take care of the neurons and the synapses. And in the presence of neuroinflammation, which is driven primarily by soluble TNF, these parents become dysfunctional. And when they're dysfunctional, they don't do their job. They don't take care of the neurons, the synapses, and that degenerative process starts. So our drug, XPro, and the goal of our phase 2 trial is to remove soluble TNF, neutralize it, and then eventually destroy it, such that neuroinflammation does not occur, and these microglial return to the state that they originally intended to do, and they start taking care, and they start doing their jobs, and they start taking care of the synapses and the nerve cells again. And that's why we believe we can potentially flatline cognitive decline, what we saw in some of our phase 1 results. So that's our view on Alzheimer's, and we'll find out towards the end of next year if that's going to come to fruition. Now, let's move on to treatment-resistant depression. So treatment-resistant depression is for patients who have failed multiple therapies for depression. If you look here at the chart on the left, you see that the number of failed antidepressants, what occurs there is those patients, as they fail more and more antidepressants, they have elevated levels of TNF, higher levels of TNF. Turns out that there was a trial that was run with TNF inhibitors, and they included all TRD patients, and the results actually didn't come out positive. But if you bisected that trial and you looked at patients that had if you enrolled patients that had elevated levels of CRP, which is a biomarker and a surrogate of inflammation, there was actually a dramatic difference between those that were treated with a TNF inhibitor and those that received placebo, as you can see with this red circle. So that gives us, and as you know, to get a drug approved, you need to show separation between the treated versus the untreated group. So this gave us really good confidence that if we can take TRD patients that have elevated levels of neuroinflammation, so they're all hot, treat them with XPro, which is a TNF inhibitor that works differently than the existing TNF inhibitors, we can show that separation. I'll remind you that we're doing the same thing with the Alzheimer's trial. We're only taking patients that have elevated levels of neuroinflammation. We know that patients that have elevated levels of neuroinflammation with regards to cognitive decline much more rapidly, and the variance that occurs is a lot less for those with elevated neuroinflammation. So that allows us to show a bigger separation from our treated versus untreated compared to the normal group. So that is XPro. I'm now going to switch gears and I'm going to talk on one slide only about our two programs in INmune. So INmune is a program that changes the phenotype of the patient's own NK cells. And it does so by interacting with the patient's NK cells such that a whole series of proteins get upregulated on the NK cell surface. There's multiple ways to do this. You can use cytokines to upregulate proteins. You can engineer it outside the body, and you can insert these NK cells. But there's issues associated with those approaches. With regards to cytokines, you're limited in terms of how you can give those to patients because they're quite toxic. And the second thing is that you have to constantly give these cytokines to maintain that status. With regards to INmune, what we see the same upregulation of proteins as a combination of cytokines, plus some additional ones related to what we call mitochondrial survival or nutrient proteins, which allow the INmune-primed NK cells to survive better in a hostile tumor microenvironment. So with MDS, this is really our goal. If you look at patients, if you took a group of MDS patients and you looked at their NK cells, if you look at the chart on the left, patients that had higher levels of activated NK cells survive much longer versus those that didn't. And this is kind of our original thinking that we want to activate those NK cells to increase survival. If you look at the chart on the right, again, this shows that patients that have, on the blue line, that have a very cytotoxic, much more active NK cell survive much longer than those in the red line, patients that don't. They have impacted NK cells. And our goal with INmune really is to take patients from the red line and switch them to the blue line, or maybe even do a little better than the blue line, but let's say do somewhere near the blue line to increase survival by changing the phenotype of these NK cells. So now let me switch from our MDS AML program over to, finally, our prostate program. So one of the reasons we chose prostate is because the large tumor, there really hasn't been much in the terms of advancements in the last 10 years. And the immunotherapies that they've tried to date have really failed. And if you look at why, what excites us about INmune for prostate cancer is that within the prostate tumor resident are already a lot of NK cells. So if you look at this chart in the box on the red, patients that have prostate cancer tumor burden have an elevated level of what we call resting NK cells. That's that bar all the way at the top. But with regards to T cells in tumor, you would want to see this bar moving to the right in the green box. It doesn't exist. So when they use drugs like TIGIT drugs or PD-L1 inhibitors, they didn't work. Why? Well, there's no T cells there. So it's hard to get an activated response when you don't have the cell that you need to make your drug work. I'm guessing, and this is purely a guess, if these T cells were off to the right, like we see with the NK cells, maybe the PD-1 inhibitors and so on might have done a better job. So our belief is that we already know the NK cell is there within the tumor, but they're just sitting there in plain sight, resting quietly. And our goal with INmune is that if we can activate these, which we've shown that we've done with our other patients, and obviously in many preclinical studies, that these NK cells become cancer-killing, what we call memory-like NK cells. And they will do the job that they were intended to do, which is to go after tumor. So this is what gives us a lot of interest in pursuing this trial, and we'll look forward to sharing data in all of these programs once we achieve them. So hope you understand the rationale in a one-slide overview of our program, and thanks for watching. And again, please comment, and we'll look forward to talking on the next episode. Thank you.